A lot of oocytes in here. So if you notice, like the size is really unique. The oocytes are huge cells. So all these little dark dots, those are normal sized cells. The oocytes are monsters. Huge, huge cells. And so up here, this little cluster right here, these are surrounded by basically little squamous cells. So these are your primordial follicles. There are tons of them. They're the smallest. So it's the only one you can really use size to do. Like all you got to do is know that there's a big cluster of really, really small for an oocyte type cells. As you move closer to the center, notice this one, that's I think a pretty good example. These are more cuboidal cells surrounding them. Single cell follicles just blown up a little bit. Same with this one here and this one here. It doesn't quite circle it perfectly. But that would be another primary follicle with a primary oocyte in it. Down in the bottom right, there's a nice stratified one. Again, still a primary follicle. Here's one right here too. So these are all primaries. There's a really nice one. Bunch of primary follicles with primary oocytes inside. So if we go deeper, we'll probably run into some different type. And on this side, it gets a little more complicated. These are all still primary. See if we can get one secondary in here. Here's one that is just beginning to become secondary. You see that white crack that's forming right here? So you got a little crack there, little crack right there. So that is like the earliest sign of the formation of a secondary follicle. I'll give you a bigger one that's a little bit more clear than that, but that's kind of what you're looking for. This breaking away, what it's forming is the corona radiata, and it's breaking away from the rest of the follicular cells. And so that's what I'm talking about. It's really a nice little white space with one layer of cell under it. But I'll try to pick one that has a little bit more significant opening, more like the drawing that we saw. And you guys in your slides do have quite a few decent examples of a secondary follicle. And I don't think there are any tertiary follicles. Oh, maybe over here. That one's kind of broken through. So it's not a great example, but this gives you an idea of the size. A mature follicle, the biggest follicle, is at least this big, usually much larger. Let's see if they have a different slide at a different time of the day. Let's see, that's corpus luteum. Let's go back here. This is the one for the PowerPoint slide. So again, just take a look at those. Primaries up at the top, or primordial up at the top, primary follicle down here. And then this one, again, just to emphasize the difference in size. What you'll see later on, taken at a different time for a different slide, you'll get these much, much larger ones where you see this very well-developed antrum in here. So that's really it for picking out the follicles versus picking out the cells. So make sure you are always being clear with yourself. Am I naming a cell or am I naming a follicle? You're probably going to spend most of the time today picking out follicles. You're not actually picking out the cells. There's only two kinds of cells here. Primary oocyte, that's primary oocyte and secondary oocyte. And again, the secondary oocyte doesn't show up until the mature follicle. Then ovulation happens. And the end of this story is the follicle actually turning into a really powerful endocrine tissue. And that's called the corpus luteum. It means yellow body because in reality, if you look at it in an unstained organ, it is quite yellow. When it's stained, it's kind of pink. So under a microscope, it won't actually be yellow. But this is what secretes progesterone. You don't need to know any of the hormone stuff. I'm just saying this FYI because I know you're doing it in lecture. I'm not going to ask you any questions about any hormones for lab. But just to give you an idea of what this does, to connect it with the story here, progesterone is coming from corpus luteum, which is going to maintain the uterine lining and prevent the woman from going through menses in preparation, hopefully, for implantation of this secondary oocyte. If the implantation doesn't happen, if it wasn't successful, the corpus luteum turns off, and then the uterine lining basically dies, and the whole process begins again. The corpus luteum degrades into number 9 here, which is called a corpus albicans, which is basically a cluster of scar tissue. Now wait, next year they're going to say it's like another endocrine organ, so who knows? But at this point, it's a chunk of scar tissue, and histologically there's some support for that. You'll see under a microscope, it doesn't even stain. It's really light, kind of like that light pink color right there. So it doesn't seem to be a very active tissue at all, whereas corpus luteum stains a lot. It's a very active live tissue. Yeah.
Does the albicans get flushed out during menses, or does it stay?
No, it hangs out there. So you have an accumulation of scar tissue over the course of your entire life. And it can be significant in terms of the total mass. Yeah.
So you just said about the corpus luteum, and if the egg is implanted in the uterus, it remains active.
Yeah.
Right? So as a person has children, do they remain active? Like, can you see the exact, can you see them over time? Like, could you see a slide of it, and like as she's had babies?
Like 20, 30, 40, 50 years? Like, you mean as someone ages? Would
Yeah,
they
like
change?
could you go back
absolutely.
and see which one was the one that expelled, the one that became the baby?
I don't know that you could match them up,
No, I know,
but you would have more the older you are. The more ovulations you have, every time you have an ovulation, you're going to produce a corpus luteum, which will degrade into a corpus albicans. So
even after it does become a fetus. And all that.
absolutely, yeah.
Okay,
So
so
even
we'll-
after that, it's got to go
It's gotta
somewhere.
degrade.
It just
Okay.
turns into scar
That's what
tissue.
I wanted to say.
But someone who's had more kids would have fewer, because during those 9 months they wouldn't have done it
Got it.
at all.
Oh, that's a good point.
So I put a line through this just to, it's in your lab manual. Objective 8 and 9 on your lab sheet talks about knowing these. You do need to know these for lecture, and you should know these because this is kind of the important part physiologically, but for anatomy lab, I'm not going to ask you hormone questions. You guys have talked about this in lecture already, right? So I just want to connect the terminology. Follicular stimulating hormone. Hopefully now you can see, oh, it's causing the development of a follicle. So that's what FSH is doing. It's binding to the primordial follicle and causing it to go and differentiate into a larger and larger follicle to go through this process. So that's why this surge of follicular stimulating hormone will cause the follicle to grow. Then there's going to be a surge of luteinizing hormone soon after that is going to cause ovulation. So luteinizing, the reason they call it that is that when you get a surge of luteinizing hormone, you produce a corpus luteum because you ovulate. After the ovulation happens, a corpus luteum is formed. So that's where that terminology comes from too. So hopefully let that remind you so that you don't ever forget when someone talks about luteinizing hormone, it's the ovulating hormone. Follicular stimulating hormone is the thing that stimulates the follicle to grow. And then down here you have estrogens and progesterones. The short story with estrogen is that the follicle secretes estrogen. It produces estrogen. That's why when you're actually going to see a huge rise in estrogen along with the development of the follicle itself. And then progesterone shows up once the corpus luteum is created. So boom, luteinizing hormone. And progesterone is going to go up, 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 up. But then if there's no fertilization, which is what this example is, it's assuming no fertilization, it's going to drop off when the corpus luteum degrades into the corpus albicans. So if we zoom in at the upper part of this picture, this is a little bit more important for this week's information because the last terms on your sheet are talking about the endometrium, talking about the different follicles. So let's take a look at the endometrium. So the endometrium, right, the most inner layer of our uterus, has two different layers. It has a basal layer that stays the same. Basal layer is the boring layer, but it's an important one. Because what it's doing is growing out a functional layer. So there's rapid mitosis happening here over time. So here we're at day 1. So menstruation occurs. Once menstrual flow reduces, we call that day 1, and then we're going to move forward in time. As you move forward in time, out of the menstrual phase at about day 5, you go through what's called the proliferative phase, which is the growing of the functional layer of the endometrium, also known as the stratum functionalis. So proliferative phase is just the growth phase, basically. Then secretory phase kicks in about day 14, and that's the secretion that's happening there, the investment or the creation. You build blood vessels, and you build what are called uterine glands. And those are both to provide excess nutrients for the implanting zygote. And then as you get to the end, right around 28 days, if no implantation occurs, progesterone drops off. And progesterone was the thing that was causing the secretory phase to happen. Once the progesterone drops off, these blood vessels basically pinch off, the functional layer dies, and you start this process over again. So menstrual phase, then proliferative phase, then secretory phase. If you look at the top of the image, it lines it up time-wise with the different phases of follicular development. So down here, when this is going on in the endometrium, this is what's going on in the ovary. And so here's this corpus luteum maintaining the uterine lining with progesterone, and then it degrades, and that's why the endometrium also degrades. So if there was successful fertilization and implantation, let's say it just happened right here. So ovulation, then fertilization, then implantation. If that happens, the fertilized then embryo at this time would release human chorionic gonadotropin. That's the thing that they're testing for during a pregnancy test. They figured that out by shooting them into frogs, and it caused them to produce a bunch of eggs. It was really cool. But now you can just pee on a stick. But it's going to tell you whether or not that hormone's being released. And that hormone will send a signal to the corpus luteum saying, we've had success, implantations occurred, don't stop producing progesterone. And so if that's successful, the uterine lining will be maintained up until the point the placenta can take over the progesterone production. And so that's kind of the key thing that has to happen that we'll talk about. Well, we won't, but next week we'll pick up that story from that point on, talking about embryological development. But in this image, we're talking about not a successful implantation. So progesterone would fall off, and the functional layer of the endometrium would slough off. All right, so next week is development and heredity. And so the heredity portion is review. So if you want to get a jump on that, do it. You don't need to be in lab. It's a lot of inheritance patterns and that kind of thing. Just basic